and welcome to Teach Me in 10. This episode is a brilliant one where we'll be talking about the important factors to consider when you're designing your RNA-Seq experiments. So without further ado, let's get into it.
There are various factors to consider when embarking on your first RNA sequencing experiment, from your experimental design to data analysis. This video will help guide you through what questions to ask yourself as you plan your experiment to achieve the most relevant and optimal results. Which RNA-seq assay should I use is one of the most common questions researchers have when beginning an RNA sequencing project. There are several options available, and your selection will depend largely on the experimental goals and objectives that you have for your project. Are you interested in gene expression profiling, de novo transcript assembly, alternative splicing, or isoform analysis? A well-defined objective will tell you which NGS platform and data you will need, and ultimately which assay is best to provide that data. This decision tree offers a good starting point to choose the best assay for your experiment. The blue boxes indicate the objectives of the RNA sequencing study, and the green boxes represent the RNA-seq assay to be performed. While several platforms are available for RNA sequencing, each has an advantage and disadvantage depending on the situation. For example, this platform comparison table can help you decide which is best suited for your RNA-seq project. Many RNA sequencing experiments utilize short-read platforms. This is due to their low cost per base pair, high accuracy, and high throughput capabilities. Long-read sequencing may also be an option for yourself, depending on if you're interested in isoform analysis or annotating a de novo transcriptome or improving a genome annotation, but these often have a lower throughput and a higher cost per base pair. For the purpose of this video, we'll be primarily focusing on short-read based technologies. Short-read sequencing can either be done unidirectionally, or single-ended, or bidirectionally, pair-end sequencing, and aligned to a reference genome or transcriptome, or assembled into a de novo transcript. Paired-end sequencing is usually recommended for most applications. This was because it provides a richer data and a longer insert length, permitting a longer library. It's also the most common sequencing type and is often the most cost-effective. It's crucial to consider the number of replicates needed when designing your experiment. A minimum of 3 biological replicates is recommended to ensure a valid biological interpretation of your results. Depending on your aims and objectives of your experiment, increasing the number of replicates to 5 or 6 per condition is not uncommon. RNA-seq is a high-throughput quantitative method, and experimental controls are vital to testing your hypothesis and reducing error rates. Including a known quantity of synthetic RNA as an internal reference can control variation between your samples and reduce batch effects. This synthetic RNA internal control is often referred to as an ERCC spike-in and can help standardize RNA quantification across different experiments. ERCC spike-ins are pre-formulated polyadenylated transcripts at a known range of lengths and concentrations. Using the read counts from your ERCC spike-in controls, researchers can help determine the sensitivity, dynamic range, linearity, accuracy of RNA sequencing experiments. And as previously mentioned, they can be used to control for technical variation between runs or batches. When preparing your samples for RNA sequencing, the samples should be collected and preserved as quickly as possible to ensure that you do not unintentionally alter the gene expression or activate ribonucleases, known as RNases, which cause RNA degradation. When working with tissue samples, we recommend to snap freeze your samples in liquid nitrogen. For cells in suspension, adding commercially available stabilizing reagents before centrifugation is recommended. High-quality, undegraded RNA has the best chance of producing accurate and information-rich RNA sequencing results. Two common sources of this degradation are heat and RNases. Ensuring that you're keeping your sample cold while processing and working in an RNase-free environment is vital to preserving your RNA integrity and ensuring the best results possible from your experiment. You should choose your extraction method with your sample type and goal in mind. There are many commercially available column-based kits for most species and tissues available these days. However, please be careful to ensure that they capture the quality and type of RNA required for your experiment. Modern RNA extraction protocols usually rely on columns or beads. However, it does not just isolate all RNA. There can always be contaminating DNA in your sample, and we would recommend performing a DNA treatment step to remove any of this unwanted DNA. Quality in terms of sample purity can be detected using UV spectroscopy and calculating the absorbance ratios. Good quality RNA will have an A260-280 ratio between 1.7 and 2.1, whilst the A260-230 ratio should be between 2 and 2.2. An A260-280 lower than 1.7 may indicate that there's protein contamination of your sample, whereas an A260-230 lower than 1.8 may indicate the presence of organics such as phenol or guanidine left over from your extraction protocol. Such contaminants can interfere with the library preparation and should be further cleaned up prior to sequencing. Ribosomal RNA makes up the vast majority of the RNA in eukaryotic and prokaryotic species. Therefore, a method to enrich your messenger RNA or deplete the rRNA from your samples must be selected. For eukaryotic species, researchers can choose between a poly-A selection, using the poly-A tail of the messenger RNA for enrichment, or rRNA depletion to enrich non-ribosomal RNAs during the preparation steps. For prokaryotic species, they require rRNA depletion methods as their mRNA is not polyadenylated. This decision tree can help guide the selection and enrichment method used in your assays. Understanding the polarity of transcripts is critical for genome annotation and novel transcript discovery, but this information can be lost during the cDNA synthesis and subsequent amplification steps of your assay. To preserve this information during library preparation, consider a strand-specific RNA-Seq method, also known as stranded or directional RNA-Seq, which determines the gene orientation that the transcript originated from on the DNA template. How much data do I need is another typical question asked when starting an RNA sequencing project. For RNA sequencing experiments, read depth is normally expressed in read pairs per sample. With paired-end sequencing, you can express this value either as the single reads or the pairs. For example, if you sequence 20 million fragments or clusters on a flow cell, with a paired-end strategy, you would generate 40 million single reads or 20 million paired-end reads. Most experiments will need somewhere between 5 and 200 million paired-end reads per sample, and general smaller genomes with fewer genes need fewer reads for gene expression quantification. For experiments where you may be looking for splicing events or rare transcripts, you may want to increase this data amount substantially. Choosing the required read depth depends on the application. This table refers to short read sequencing technology only. When performing deeper sequencing with minimal input material, consider using UMIs or unique molecular identifiers. PCR does not copy cDNA libraries with perfect fidelity. It's often biased, amplifying some molecules more efficiently than others, and it can also introduce errors or mutations. By tagging the original cDNA molecules with a UMI, all PCR copies of it will be identified by that same barcode. After sequencing, bioinformatic tools can remove the PCR duplicates from the dataset. PCR and sequencing errors can also be identified and corrected using the UMIs. The resulting data is then all ready for gene expression analysis. The final bioinformatic pipeline and tools you choose will rely on your final experimental objectives. For short-read RNA sequencing experiments, however, the typical analysis pipeline looks as follows. Post-sequencing, read quality is assessed using tools like FastQC. This helps to ensure that there was no serious issues with library preparation, sequencing, or contamination of your libraries. The reads can then be trimmed to remove any low-quality bases that appear at the start of sequencing reads. A filtering step is then further applied to remove any low-quality reads from the dataset. The reads are then either aligned to a genome or a transcriptome of your species of study. Following the alignment, the number of reads per transcript are counted before moving to tools such as DE-Seq2 for differential gene expression analysis and more advanced analysis. We hope you found this video helpful. For a deeper dive into RNA-Seq, download our ebook, A Guide to RNA-Seq, below. It provides more information about each of the topics covered here today, as well as tips to ensure that you get the most out of your RNA-Seq experiment. Thank you for watching.
Thanks so much for watching this episode, which was brought to you by GeneWiz Multiomics and Synthesis Solutions from Azenta Life Sciences. GeneWiz from Azenta provides leading global genomic services, including Sanger sequencing, next-generation sequencing, gene synthesis, molecular biology, bioinformatics, and GLP regulatory services. With multiple locations worldwide, GeneWiz from Azenta Life Sciences is the preferred partner for leading academic, pharmaceutical, biotechnology, agricultural, government, and clinical institutions for easy access and support. So make sure you like and subscribe, and we'll see you again very soon.